I want to talk to you now about crossing over, and that really reinforces again this idea of linked genes that influences the phenotype ratio. Crossing over is an exchange of segments of chromosome between homologous chromatids during meiosis 1. So during meiosis 1, we have our homologous chromosomes. Remember, they're replicated, so we've got 4 chromatids, and they align together as a tetrad. Each one of these is a chromatid, and I'm going to have different colour for the homologous chromosomes. So that's one chromosome, and the homologous chromosome is going to be a different colour. Now I'm deliberately going to be touching here. So they're homologous chromosomes. What does that mean? Well, they've got the same genes, but perhaps different alleles of those genes. So they're homologous. Each one of them has replicated, so we now have 4 chromatids. Now they align together during meiosis 1. And so the first part of the process is called pairing. We have pairing of the homologous chromosomes. The second thing that happens is that we have crossing over. Now, crossing over is actually the process of literally crossing over here. So we've got one chromatid here in physical contact with one of the homologous chromatids, and we've got the same thing down here as well. So in that process, we actually can have a swapping of some of the chromosome. A segment of the chromosome actually literally moves from this chromatid onto this chromatid. So I'm going to see if I can demonstrate that. Okay, so this segment of chromosome here now attaches here, and the segment that was on the other chromatid now attaches here, and it's the same thing down the bottom as well. Now this part of the process is called recombination, and it's the last part of the process. So basically it's joining on, so that that chromosome segment combines or recombines with that chromatid, so it actually joins onto that chromatid here. Recombination. So then at the end of meiosis 1, where these 2 homologous chromosomes separate, we now have some of each of the chromosomes. So some of the mother's chromosome, some of the mother's genes are now with the chromosome from the father. So what crossing over allows is it allows that the offspring has a different genotype to the parents. So essentially, if we've got 2 genes that are on the same chromosome, they should only have the parental genotype. But with crossing over, we can have a genotype that's different to the parent. And that genotype we call the recombinant genotype. So I'm going to write that down as a note. But the take-home message here is that crossing over provides variation. Crossing over means that we have potential to have a genotype that's different to the genotype of the parents. It means that the 9331 dihybrid cross phenotype ratio is another reason why that might not actually work out as predicted, as theoretically predicted. Okay, come here. Alright, so if 2 genes are on the same chromosome, the offspring should have the parent's genotype. But with crossing over, the offspring can have a genotype that's different to the parents. There you go. Does that make sense?
Yeah.
Good.
Good.
Go to bed.